Chapter 6, Cell Division. So what this chapter is about is how do you grow? How do you grow, develop? So if you're a fetus in the womb, growing, getting larger every day, your cells have to divide for that to happen. If you cut your finger, when your body heals itself, new cells are being produced. And that's happening throughout your body all during your life. Well, also the production of sperm and egg for sexual reproduction, all of these things involve some type of cell division or another. The cell cycle is basically the circle of life as far as cells are concerned. The cell spends a large amount of its time growing, getting bigger, but then it gets too big and it needs to divide. So that's what these different stages are. You can see here, it's kind of like a clock. You spend most of the time, the cell spends most of its time here in G1, S, and G2. So that's from about 9 o'clock until about 6, all the way around the clock. And then when it's time to divide, it goes through mitosis, which is another way of saying cell division, and then eventually cytokinesis, right? So interphase is what most of the lifetime of the cell is spent in. Then when it decides that it needs to grow, that's called G1 phase. And if it decides it's time to reproduce and divide, it goes into S phase, which is when the DNA is duplicated. That's a very important moment in the cell. S phase stands for synthesis. The DNA is duplicated. Remember that. G2 phase, it just makes its final preparations and then it goes into cell division. The G in G1, G2 stands for gap. It's not as important as the S phase, but they're growth periods. They also serve as checkpoints. We'll talk about that in a little while. But at the edge of each of these different colored lines are checkpoints where the cell analyzes itself and decides whether it should grow and divide or not. And something bad happens if it goes through those when it shouldn't. Interphase is the time that the cell grows, and it spends most of its life in interphase. G1, S, and G2 phase. Briefly, when it's necessary, it goes through cell division. And cell division here is shown as mitosis, but it can also be meiosis. Let's dig in a little deeper. Number one, you can see the chromosomes in the cell. And then when synthesis happens, they double because it's like when you're packing your house to move, you have to prepare to move. And then at the checkpoint, when it's going to go to the next stage, you can see those chromosomes are visible. And then it actually lines them up and divides and splits them right down the middle. If you look very carefully, these chromosomes look different than these because they have been split and divided and partitioned into two brand new cells. Some cells do not complete the cell phase, the energy zero. That just means they're endlessly going around and around, but never dividing. It's a non-dividing state. It can last for a few days to the lifetime of the organism. So G0 is just when it doesn't divide. And as soon as it goes into division, then it's going to properly go through G1, S, G2, mitosis. And here's another word that's important, cytokinesis. That's the division of the cell. Now, you might say, well, wait a minute. I thought mitosis was the division of the cell. It is, but technically mitosis is just the division of the nucleus. You double your DNA, you split your DNA, you build new nuclei around that, and then only if you split the cell at that point do you go through cytokinesis. So technically mitosis is just nuclear division, and technically cytokinesis is just cell division, but they usually go together. Most single cell organisms use sexual or use cell division to reproduce offspring through asexual reproduction. This generates clones, offspring that are genetically identical to the parents. If you cut your finger and you heal, the cell division that's happening there is mitosis, which is asexual. It's not until you make sperm and eggs that you use sexual reproduction, which is also known as meiosis. And then bacteria, they don't do any of that. They do what's shown here. They just do simple binary fission. They have one circular chromosome, they double it, and they partition it into two new cells. They split that mass into two new cells, each which has two which just has a single chromosome, and those are called two daughter cells. It's important to understand they're called daughter cells. We'll use that term a lot. Eukaryotic cell division is more complex because our DNA is more complex. Bacterial cell just has a singular circular chromosome. We have 46 and they're linear. So it's much more complicated. Eukaryotic cells undergo two types of division, asexual and sexual. Asexual is mitosis, and that's just growth. You produce clones, just like when your cell heals in your finger cut, those are clones of the cells that were there before. Sexual reproduction, also known as meiosis, is the production of sperm and egg. So the result of mitosis is a new skin cell or a new brain cell, but the result of meiosis is a sperm or an egg. Watch the video when you get a chance. Mitotic division or mitosis is a type of cell division in eukaryotes. It does not happen in prokaryotes, so it only happens in plants, animals, protozoa, fungi. It does not happen in bacteria. They use binary fission. The stages of mitosis can be summarized by the acronym PMAT. PMAT, prophase, metaphase, anaphase, and telophase. You should know those and what happens in each one. So here's interphase. Nothing's happening there except for growth. The chromosomes aren't visible. If you were to try to draw them, you would just be drawing spaghetti. But then finally, at the very beginning of mitosis, and we enter prophase, now the chromosomes are visible in a microscope, and you could draw them, and you would draw them like little butterflies. Then at metaphase, what happens is the chromosomes line up in the middle of the cell along the metaphase plate. It's an imaginary line right down the middle, and they prepare to divide. In the next phase, anaphase, the chromosomes split in their middle. Their middle is called centromere, and they split at the centromere. So now if you look at this red chromosome, it has split into two pieces, and those pieces are called sister chromatids. The same with the blue, the same with the other red, and the same with the other blue. Then the nuclear membrane reforms in telophase, and the cell pinches. It forms a cleavage furrow, and it pinches, and then it becomes two cells, goes back into interphase with the nuclear membrane fully formed, and now the chromosomes disappear because they become all spread out, and you can't see them anymore. During interphase, the cell's DNA is not packed tightly, but remains loosely associated with proteins. The chromosome is replicated during S phase. When that replication happens, you go from one strand to two. And now if you look here, you can see the two strands. They are each called a sister chromatid, and they pinch in the middle and attach, and that's called the centromere or the central pinch. Watch the video when you get a chance. Now, if this doesn't work properly, we have cancer. At each intersection between G1, S, G2, M, and cytokinesis are checkpoints. They're proteins, and they tell the cell to stop, take a minute, and evaluate yourself. Do you want to go to the next stage? If not, you just stay in G1. You may stay there indefinitely. Now you're in G0. But if it is time to move on to the next stage, that checkpoint will let you go. They're kind of like little gates, and they open and close. But if they're broken, that's when cancer happens, because cancer is your body's natural healing mechanism gone horribly wrong. So instead of growing and dividing when it's supposed to, it just grows and divides, grows and divides, grows and divides, grows and divides. It doesn't know when to control and stop itself. So instead of getting a nice little organ formed the way it's supposed to, and then healing when it happens to, and then stopping, it just forms these uncontrolled cell growth, and that's where you get a tumor. Tumors are mass of rapidly runaway cells. They're benign if they stay in one site. That's why someone could say to you, it's a tumor, but it's not cancerous. That is called anchorage dependent. Angiogenesis is how it supplies itself with new blood vessels, and then it is cancerous. Those are called malignant. And then if they spread, that's called metastasize. All right, now we move on to meiosis. Meiosis is different than mitosis. It has a different goal. Mitosis is just to make clones. If you just want to grow and heal, get bigger, then you do mitosis. But if you want to produce sexual offspring that are unique and different, and might have unique characteristics, you have to go through meiosis. So what happens is you take your DNA and you cut it in half, creating a sperm and an egg, and then when you fertilize, you bring it back together again. So we have to understand these two new vocab words, haploid and diploid. Haploid is half of your DNA, whereas diploid is both. Now, how can you remember that? Diploid has a D for double, and haploid has an H for half. So in humans, if we have 46 chromosomes, that's our diploid number, 2n equals 46. But if we split that in half during production of sperm or egg, we go to haploid, half, which is n equals 23. The body cells are diploid, but the sex cells, sperm and egg, are haploid. And then when they come together, they restore the diploid number. Fertilization is when they fuse the sperm and the egg. You get a zygote, which is a diploid cell that eventually becomes a fetus. Now, we have pairs of chromosomes. That pair is called a homologous pair. And in mitosis, when you drew all those chromosomes, they don't divide and split, they stay together. The sisters split, but not the homologous chromosomes. In meiosis, this is very important. It's the most important thing in this chapter. In meiosis, the homologous chromosomes separate. That is the most important thing. DNA is replicated during S phase in meiosis, and then we have two stages, meiosis 1 and meiosis 2. But the good thing is they involve the same phrase of PMAT. It's just that it's prophase 1, metaphase 1, and then when you get to metaphase or meiosis 2, they're now called prophase 2, metaphase 2, etc. Now, the first stage, meiosis 1, you go from one cell to two, and what you do there is you split the homologous chromosomes. And then it goes through another division called meiosis 2, and there now you're splitting the sisters. So when you're drawing meiosis and practicing it, making sure you understand it, if you can just get this stage here set up properly, once you get to here, then it becomes just like mitosis, because now you're just splitting the sisters. But what's fascinating, though, is at the very end, you have four cells rather than two, and each cell is unique. It doesn't have the same exact chromosomes as each other, and it does not at all look like the parent. And that's what makes sexual reproduction amazing, is that the offspring are all different. Meiosis 1. The two members of each homologous chromosome pair and are separated. The sister chromatids stay together, which is different than in mitosis. The produce is haploid cells. So immediately at the end of the first meiotic division, you have two cells and they're both haploid. Now let's look and see what that means. In this cell, we have a total of 4 chromosomes, too big, too little. But over here, we have a big and a little. That is a haploid cell. We've lost half the chromosomes. Meiosis 2, the homologs are split. Now what we have to do is split the sisters. So if you look here, you've got 2 chromosomes, but they both have 2 sister chromatids. We have to split those. So now when you count it, you count 2 chromosomes, but notice something else. Each only has 1 chromatid. And if you look really carefully, they're all different. Very helpful video. Pay attention to when the ploidy level changes, when it goes from 2n to n. In meiosis 1, that's very important. The most important thing in this chapter. So the result of it is that they're similar but not identical. Half the organism's DNA comes from a different parent. Meiosis and fertilization maintain constant chromosome number while allowing for genetic diversity. Here's how meiosis creates genetic diversity. 2 ways. 1, crossing over. Number 2, independent assortments. When you draw it, you'll start to see what that looks like. You could have had them line up in different ways. So for example, here it was red on the left, blue on the right. Below that, you had the same configuration, but it didn't have to be that way. You could have lined them up so that the reds were not on the same side, and that's called independent assortment. Crossing over is where pieces get exchanged. That's why you'll see like these little pieces of blue and red in different places. Genes break off and recombine with genes that you've never combined with before. For example, if on this chromosome you had blue eyes and blonde hair, over here you had darker features, by recombination, now you can break those traits apart. So you could get an offspring that had blue eyes but dark hair. That is what happens during recombination. Independent assortment is just reshuffling the deck a different way. And again, it's another way of creating diversity. You'd have to draw it to see it, but you can see it here a little bit. Notice that we line up the reds on one side, the blues on the other, but here just randomly they organize themselves the opposite. It's not reds on one side, it's red, blue, and over here it's blue, red. So the result would be different offspring in the bottom. Just look through the offspring at the bottom and you can carefully see that. Now, the most important thing to remember is that mitosis is growth, healing, and of course, if it goes wrong, it's cancer. Whereas meiosis is for sexual reproduction. You use it to produce sperm and egg. So down here, each one of these would be either a sperm or an egg. And then when a sperm and an egg, these two get back together again and fertilize, now you restore everything. You get back to having a total of 4 chromosomes, a red and a blue of each type, and it looks more like the parents. But even though that's true, if you look very carefully, the mixtures are different. And that's why sexual reproduction is better, because you get more diverse offspring, which you might need in a changing environment. And that is chapter 6.